Good morning, all. I'm going to go ahead and I'm making a recording of the PowerPoint for Chapter 1.
Welcome to microbiology. All right, so we are starting with Chapter 1. Chapter 1 takes a brief look at the history of microbiology and covers some other important concepts. Here's a list of the names that you will need to know for the exam. So let's go ahead and go through these. This is an animation that you should watch on Francisco Redi. Francisco Redi actually experimented using cuts of meat to help to prove to people that organisms couldn't just spontaneously generate or arise by themselves. So here's a little bit of an overview. Francisco Redi, his experiments had to do with decaying meat. And basically, he was trying to prove that flies couldn't just develop from meat unless the meat had been exposed to flies. And this is all because of Aristotle. Aristotle had put forth a theory of spontaneous generation, that life could just come from nothing. And obviously, Aristotle, being such a famous scientist, this mistake that he made, or philosopher, this mistake that he made, took a long time for people to disprove that he was wrong on this. So here's what Francisco Redi did. He had 3 flasks. He left one unsealed so flies could get in, and you can see they lay eggs, which then hatch to form more flies. He sealed a flask, and he covered one with gauze. And as you can see, the only place where the meat grew new flies was the flask that was unsealed. Next scientist is Antony van Leeuwenhoek, and he invented the first microscope. So he basically, and his microscope really looks a lot more like a magnifying glass, as we'll see in the next picture, but he used it to examine water, and in the water he would see animals, fungi, algae, protozoa. So you started to see microorganisms, and he didn't call them microorganisms, but by the end of the 19th century, that's what they were called. And this is what his microscope looked like. And these are some of the images that he could see under the microscope. All right, so different types of microorganisms. You might brainstorm on this for a minute. We're going to cover a wide variety of microorganisms in this class. Generally, when we think about microorganisms, we think about bacteria, fungi, protists, viruses, and then sometimes you might think about things like algae in that category. We're going to cover prions, and then we'll talk about helminths, which are worms that cause disease, although most of those aren't microscopic, some are. And so those are some of the different types of microorganisms. Eukaryote versus prokaryote. So hopefully this is something you remember from previous biology classes. Eukaryotes have nuclei and prokaryotes don't, right? So prokaryotes are a lot smaller because they are not as well organized. They don't have membrane-bound organelles like nuclei. And so they're much smaller and simpler, and those are our bacteria. Eukaryotes are a lot more complicated and bigger because they have so much better organization. So we are eukaryotes, and we'll be seeing these two in detail coming up in Chapter 3. All right, so here are some of the groups of microorganisms that I had mentioned earlier. And I just realized I had left archaea off of the list. Archaea are similar to bacteria in that they are prokaryotes and they are single-celled and tiny, but we're going to see a bunch of differences. They're an older group and they don't have any pathogens in them, meaning they don't cause disease. Carolus Linnaeus is one of the first people to start classifying things. Now, he didn't use these 6 categories, but those are 6 of the main categories that we use now. Okay, so to go through different types of microorganisms, bacteria and archaea are our prokaryotes. They're unicellular and lack nuclei. They're small. They're found everywhere. They reproduce asexually. Bacteria contain peptidoglycan in their cell walls. Almost all have cell walls. There's one group we're going to see that doesn't. And archaea don't have peptidoglycan. They have polymers. And everything on this slide, you're going to have to know many times this term. So this is something you should start to learn. We'll talk a little more about peptidoglycan. It's basically a sugar protein that bacteria have. Archaea doesn't have that. They have other types of molecules that make up their cell walls. But this is really important information for this class. So start to learn everything on this slide. In this picture here, you can see two cheek cells. These are human cheek cells, and the nucleus inside the cheek cells. And all those little tiny dots are bacteria. So those are prokaryotes, so you can just get a feel for the size difference between a prokaryote and a eukaryote. And here's the classification scheme. Now this is kind of, I feel like, upside down, because I usually start at the bottom and work my way up. So I kind of like it when it's flipped over, but it'll work for us. At the bottom, the first group are the domains. These are the biggest groups, and there are three. We're going to learn all about these. There's bacteria, the eukarya, and the archaea. And again, these are three groups you're going to really need to know as we go through this term. So domains are really big groups of organisms that have some basic characteristics in common, but they don't have a lot in common. But they're big groupings, and then we start to split those up into smaller groups. So in the domain eukarya, which are all the eukaryotes, there are several kingdoms, and one of those is the animal kingdom. Okay, and when we go into the kingdoms are made up of several different types of groups. And one of the groups in the animal kingdom is the chordata, or the chordates, which means they have a backbone. There are other groups in the animal kingdom as well that are the animals that don't have backbones, which you might remember are things like sponges and some of the different types of worms. And so those would be what some of these other little circles are indicating. There are other groups in the animal kingdom. We'll actually see several of the other groups later this term. Anyhow, each of those groups in the animal kingdom are called a phylum, and the chordates, the animals with backbones, is one of those phylums. And then all the different animals that have backbones, there are many different groups of. There are different types of fish with backbones, birds, amphibians, reptiles. So each of these different rings represents a different group of organisms with a backbone. We call those classes. So we're in the class Mammalia, or mammals. And then the group of mammals is made up of different groups which are called orders. So we are in the order of, well, this isn't us. This is where we changed to a different group, but we were in the same class as mammals. Now we're looking at this panther, so he's in the order of carnivora, right? We're in Hominidae, I think is our class, but our order, but anyhow, carnivores. So all the carnivores are in this order. And then the carnivores are split up into families, and one of the families is a cat family, which is the Felidae. And then there are different types of cats groups, and one genus is the Panthera, and the different groups of organisms in genus is called species, and Panthera pardus is this particular jaguar. These are the levels of classification, so you will need to know them: domain, kingdom, phylum, class, order, family, genus, species. Right, another group of microorganisms are the fungi. So fungi are different from the bacteria and archaea in that they are eukaryotes, so they are much more like us. They obtain food from other organisms, which is sometimes called heterotroph if you get your food from other organisms. They have a cell wall. They include molds, which grow with filaments, or what are called mycelia or hyphae, and yeasts, which grow by buds and are small circular structures. Here are some images of what fungi can look like. Protozoa. So protozoa are eukaryotes that are single-celled. They tend to be similar to animals, although there's some variation there, and they need nutrients, and their structure, their cells and things look a lot like animal cells. And they're usually in the water, and they tend to live freely. Some live in animal hosts, meaning that they're parasites. They are generally asexual, although there's a little bit of sexual reproduction, and they can move. Here are some pictures of different types of protozoa that we'll see. The top one here is an amoeba. Amoebas move by pseudopods. The second one here is a ciliate, it's called, and it has little cilia to help it move. And the third one here, you can see the flagella on these. This is actually a euglena is the type of protozoa it is, but you can see its flagella. Algae. So algae are also eukaryotes. They can be unicellular or multicellular. They do photosynthesis. And we name them based on basically their appearance, by what pigmentation or what color they are and what their cell walls are made of. Here are some different types of algae. Other organisms of importance are like parasites and viruses. So parasites include the different types of helminths or worms, parasitic worms, for example. We'll be studying those. We'll also learn a little bit about like ticks and things like that that can spread disease. And viruses. Viruses are really important, as you know, from recent history here. And we'll be spending some time on viruses. And here's a parasitic worm. And here's an image of a virus. Anytime you see a virus that looks like this, it's so fun and cool with the little legs and looks so alien-like. These are actually bacteriophages, or viruses that infect bacteria, and we call them bacteriophages. Our viruses never look quite like that. Right, next famous scientist is Edward Jenner, and he is really fun, and I love this video, and I really would like you to take a little bit of time to watch it. Edward Jenner is a fascinating scientist. He was a doctor, and he discovered the smallpox vaccine, first vaccine against smallpox. And so this video helps you to understand what he did and how he did it. Golden Age of microbiology. This is just a time period, 1800s to 1900s. And in this time period, we're still working on disproving this idea that spontaneous generation occurs. We're still trying to prove that it does not. Learning a little bit about fermentation and about diseases. Okay, so even though we had seen Reddy's experiments earlier where he proved that flies couldn't come from meat, there were still a lot of scientists and people who believed that microorganisms could appear from non-living matter, even though bigger things couldn't. And so a lot of work still had to be done. This is Louis Pasteur. He's the person who helped to finally prove that microorganisms couldn't spontaneously generate either. And he used swan-necked flasks. All right, and you saw in the Reddy video, I think that also covered how Pasteur's experiment went. But here's just basically a brief overview. He used what are called swan-neck flasks, which means they had a vent in them. And that vent would help to capturing microorganisms before it could get into the broth. Okay, so he basically showed that there had to be microorganisms getting into the broth for it to be contaminated and growth to occur, and that microorganisms couldn't just spontaneously grow in the broth. So, because before that, people believed air could actually cause fermentation to occur, and other people thought organisms caused fermentation to occur. And so Pasteur proved organisms cause fermentation to occur. Here are his experiments. I'll let you read through these. Don't feel you need to memorize the steps. You can just understand that Pasteur helped to prove how fermentation works. And he basically helped prove that you needed microorganisms for fermentation to occur. So he started to use his research to understand how to better treat foods that we eat. So he developed pasteurization. So pasteurization is heating up liquids enough to kill the bacteria in them. And this basically began an entire field of industrial microbiology where we intentionally use microorganisms for manufacturing things. A few other famous people: Ignaz Semmelweis. a super fascinating person. I think I have an article posted about him. He was a doctor, and he helped to prove that hand washing was really important. And we'll see some more slides on him in a minute. And then John Snow. We'll get back to John Snow later this term. He was the first epidemiologist. Epidemiologists study the spread of diseases. John Snow was the first epidemiologist because he helped to trace and figure out what was causing a cholera outbreak in London, and he used maps and data to do that. So here's Ignaz Semmelweis. Basically, this is also in London in the mid-1800s. When women went to have babies, there were basically two clinics. In the mornings, medical students, first clinic, medical students would be the ones to deliver the babies. That group had really high mortality rate of moms and babies. And what they would do is they would actually see the cadavers first and assess the cadavers, and then they would go deliver babies. Well, at that point in history, people believed that gentlemen's hands were always clean, and so they didn't need to wash their hands. The second clinic had much lower death rates of babies and moms, and midwives delivered these, and so these weren't medical students who had just seen cadavers. So Ignaz Semmelweis really helped to prove that you needed to wash your hands. And so these were basically his experiments. You can see he got this clinic to institute washing hands with a chlorine hand wash, and all of a sudden death rates dropped drastically. I mean, sometimes some months up to 30% of the patients were dying. And so he helped to prove that you needed to wash your hands. Florence Nightingale, another famous contributor to the field of microbiology. She was really important in helping to keep wards clean. She really believed in using clean dressing and clothing and cleaning to help lower infections and disease. And Joseph Lister, you've all heard of Listerine. He basically invented what's called Lister's antiseptic technique. He was the first person to spray like instruments and clean instruments and with like an alcohol type mixture to help get rid of any contamination. So he sterilized his instruments. He was a doctor that had much lower infection rates as a result. Paul Ehrlich created the first antibiotic, the first man-made one, and it treated syphilis. It was called Salvarsan, and we'll learn more about syphilis later this term. Syphilis is deadly. This antibiotic was pretty crazy because it was made from arsenic, but it did help to treat syphilis. And there's a picture of Robert Koch. Robert Koch was super important because he gave us Koch's postulates, which you'll need to know at a later date. And he used, basically he developed this field of etiology, which is trying to figure out what organisms cause disease, like what the disease is caused by. He worked with anthrax, and he also helped establish a lot of our properties in microbiology. So he did simple staining techniques. He took some of the first photographs of bacteria. He estimated the number of bacteria in solution. He used heat to sterilize. He used petri dishes. He was a microbiologist, very much like you are going to be in this term today. So Robert Koch was a really important sort of establishing the field of microbiology. We'll be using a lot of his techniques. And so here is like a petri plate, or these will often be called petri dishes or petri plates throughout the term. They're covered with an agar, so this is like what you're picking up in your lab kits. And bacteria can grow on them. And so Robert Koch studied bacteria growing on them. You can see, like we see here, every bacteria looks a little different, right? We'll be talking about some of the different shapes and the way bacteria look throughout the term. Robert Koch also discovered Koch's postulates, which you'll eventually need to learn. You won't have to learn them for the first test, but it's a 4-step process. Basically, the suspected cause causative agent. Whatever we think is causing the disease has to be found in every case of the disease. Then you isolate it. So you isolate like the bacteria, and then you introduce it into a healthy host, into a healthy organism, and that organism needs to get the disease, and then you re-isolate it. This is called Koch's postulates. So there you go. For example, here's a diseased organism. It has a suspected agent like a bacteria or something that causes the disease. Then they take it and they inoculate it into a healthy organism, a healthy mouse. And if the healthy mouse gets sick and they can re-isolate it, then you know that it's probably that bacteria that caused the disease. Gram stain. So Hans Christian Gram discovered the Gram stain. We will be learning all about the Gram stain. It is one of the ways we identify bacteria. And basically we can stain bacteria based on their cell walls, and they're either going to appear red in color or dark purple, and this can help us figure out what type of bacteria it is. And these groups of bacteria also respond to different types of antibiotics, so it's important information to know. Alexander Fleming, early 1900s, he discovered penicillin, and penicillin comes from a fungus called penicillium, and he was just basically playing around in the lab and noticed that there was this fungus growing on his plates that would kill off bacteria. And Francis Crick, Rosalind Franklin, and James Watson, Crick, Franklin, and Watson discovered the structure of DNA. Woese and Fox, Carl Woese and George Fox, are important because they basically changed the way we group microorganisms or organisms in general, not just microorganisms, and they came up with our three-domain system: bacteria, archaea, and eukarya. You'll notice eukarya here is spelled with a C. Usually we spell it with a K. But three domains is basically what Woese and George Fox developed. And then there's just some fun information on these last slides about some of the more modern topics in microbiology. We do recombinant DNA technology, gene therapy. These are all groups of microbiology. All right, microbiology studies also how microorganisms help out in the environment. They can do bioremediation, which means to help clean up the environment. They help with recycling. They obviously cause disease. And then there are a bunch of different fields. Yes, my dog's barking in the background. You can just read about, you don't need to know these for the test, but I left them in for fun. Serology studies blood serum. Immunology studies your defenses against pathogens. Chemotherapy studies the discovery of antibiotics. And that's the list of names that you need to know for the first test and what all those scientists did. All right, and then just kind of some fun slides to look at so you can just kind of see and get a feel for what microbiology is. These are all the different subjects that get covered in microbiology and what we call the names of the topic. Like somebody who studies fungi is called a mycologist, or that field is called mycology. And here's another list of some of the other fields of microbiology. Microbiology is a huge area of study. All right, and lots of fun stuff to go with it. And then just some of the fun uses for microorganisms. I don't know if you know it, but your laundry detergent, for example, is full of enzymes that have come from bacteria. A lot of the vitamins that you eat have come from bacteria. Even like your drain cleaners often come from bacteria. And then a lot of foods that we eat, whether it's bread or yogurt or soy sauce, all rely on microorganisms for production. Right? And other famous scientists that won't be on the test but are interesting to take a look at some of the other important discoveries that have happened. All right, and here are the steps of the scientific method. In the scientific method, you ask a question, do background research, construct a hypothesis, test your hypothesis by doing an experiment, analyze your data and draw conclusions, and then report your results. Was your hypothesis correct?